And get started. So we're going to move on today. We were talking about hemoglobin before and molecules essentially binding to other molecules, right? So hemoglobin was it's the main example that's in all the textbooks. It was the one that was best studied first, although lots of things have been really well studied at this point. So right, hemoglobin, and then we talked about antibodies, but we really had our focus on proteins that were binding to some sort of ligand. And now we're going to move on and talk about proteins as catalysts. Okay, so proteins as catalysts. And so when we think about that, proteins as catalysts, we think about them, we call them enzymes. And that's going to be our focus today and from now to the exam and then past the exam as well. All right. So for those of you that are medical, pre-med or pre-something and like gross stuff, here's an example. I told you I was going to talk about this surgery my dad had where he'd gotten a blood clot and then the clot had gotten in his lungs and it had kind of grown out in there. And this is an example of this. So this is not it's not his. I meant to ask the surgeon, but I was kind of traumatized when he was having this surgery. And I've now had surgery and been in the hospital. And I now actually will say I was probably more traumatized than he was because having had the experience of being in the hospital, you're just you're focused on you. It's not complex. But anyway, other people may have had different experiences. But what he had was chronic, we'll break this down, and you won't need to be tested on this, I'm going to make sure I'm spelling it correctly. So because I was traumatized, I can't ever remember this at all. I'm blocking it all out. Hypertension. All right, and I'm going to subject you to my writing here. So if we want to break this down, what does chronic mean? Long-term, ongoing, right? So chronic we could think about as ongoing. Let's leave thromboembolic. It's a blood clot. Pulmonary, lungs. Hypertension, high blood pressure. So high blood pressure as a result of a blood clot in the lungs and ongoing, and in other words, it didn't clear up based on various let's say non-invasive treatments. And so, like I said, the way that the surgeon actually came out after 7 or 8 hours of surgery. Was surprised. We didn't think it all went well, even though nobody had given us any updates. But showed us a picture of something that looked like this. And essentially, what they do is they stop the heart for this. They pull the heart up so they can get at the lungs. And they do this for several times because they don't want somebody on the heart-lung machine too long. So they pull it up, they dig some stuff out, they pull it up, they dig some stuff out some more, and you end up getting a blood clot that kind of looks like that blood vessels that might go into the lungs. Okay? So why am I telling you about this? Besides, might be cathartic a little bit. I'm telling you about this because this is something related to enzymes, okay? And it's related to the enzyme that we're going to spend the most time talking about in here, or the class of enzymes, and those are serine proteases. And serine proteases are involved in lots and lots and lots of things, and one of those things is the blood clotting cascade. So this is the blood clotting cascade. Okay, and the reason why I'm showing it to you, and I'll blow up a little bit more, is because it is mostly made up of serine proteases. All right, so blood clotting is this balance, right? You don't want to clot too much and you don't want to clot too little. If you clot too little, you bleed to death, okay? And there's all sorts of internal damage that just kind of regularly happens to you. And if you don't have good blood clotting, you'll actually bleed to death probably internally first. But if you clot too much, then you're going to get blood clots that form, and they're going to go places you don't want, like your lungs or in your brain or next to your heart, and various things, and you'll end up with a heart attack or an aneurysm or these blood clots in your lungs, which are more rare. So let's go ahead and look at this just really quick. And so the way this happens, you won't need to know this either, but it's a good example of where blood clot or where serum protease is important. All these factors, you may have heard of blood factors before. These are just boxes here, but these are enzymes, and they're actually specifically serine proteases. So it's a serine protease. Protease means that it's going to be cutting a protein, right? Okay, so it's going to break a bond in a protein or catalyze that reaction. We're breaking a bond. How are we probably breaking a bond? Hydrolysis. We're going to add water across it, okay? It's going to be more complicated than that, but in the end, what we're doing is we're adding water across that bond. And so what you do is you have some sort of damaged surface that's going to cause a signal to be sent, and that's going to cause factor 12, which is a serine protease, to be cut or activated by, I actually don't know what these things are, but it gets activated, and now it's an active serine protease. It cuts factor 11, that is a serine protease. That then cuts factor 9, that is a serine protease. And all of these things become active after they've been cut in some place, or hydrolyzed bond. And then we have what we factor 9A, then we can have factor 8, and then that's going to, or this is actually not factor, I don't know what factor 8 is doing here, but factor 9 is then going to activate factor 10. It's a serine protease, it's going to cut it. Okay, and then finally down here, there's another pathway over here. Finally, we're going to take prothrombin, and we're going to cut thrombin, and then thrombin can come over. See, this is serine protease. Thrombin can come over and cut fibrinogen in such a way that it will make fibrin that can make your cross-linked clot. Okay? So probably didn't get what I want you to get from this is it's a giant pile of serine proteases, and one cuts the next one in. That's the way the cascade gets active, is one enzyme activates the next enzyme activates the next enzyme. Okay, and this is one of those things where it's a cascade, so there are fewer of these than there are of these than there are of these. That makes sense? So one of these guys cuts, I don't know, 10 of these, and then one of these guys cuts 10 of these, so we're getting a 10-fold each time. I made the 10-fold up, okay? But where you have damage, you want all this stuff to be activated really quickly so you get a clot so you don't bleed too much. Questions in general about that? If you think about some of your blood clotting drugs, so Xarelto is an inhibitor. So if you've heard of Xarelto, I'm going to go with that. But Xarelto is an inhibitor of this serine protease. Okay, so somebody who's taking Xarelto is going to get that. It's a newer drug, okay, and I'm not sure how often my dad has to take it, whether it's once a day or not. You may have heard of the Coumadins or warfarin. Okay, that's the old type of drug, and I didn't look this up ahead of time. I'll look it up before. We'll talk about it in more detail later. But there's a bunch of these blood clotting factors for those Coumadins that need a glutamate, needs to get another carboxylic acid put on the end, right? So glutamate has a carboxylic acid on its side chain, and yet another one gets stuck on it. So now we've got a bicarboxylic acid on the end of that, and that makes them more active. It's like 3 or 4 or 5 of those. And so that's the way warfarin works, is it inhibits that formation of that. So the activity is very different. If you are, and the way warfarin works is it does that by, I'll talk about it later when we get to vitamins, okay? But it's different. It's more complex, and you may have heard that if somebody is taking warfarin, you have to eat kind of the same stuff all the time, or you're going to need, it's going to change how much warfarin you need. You either need more or less, because what it's doing is it's interfering with how much your production of vitamin K. Okay, anyway, interesting stuff, but what we want to take away for today is these guys are all serine proteases. So serine proteases are important. So let's talk about how enzymes catalyze reactions. So I want to remind you, I've drawn this picture before, when we have a protein binding to something or catalyzing a reaction, I like to picture them as Pac-Mans with small mouths. Okay, in general, you have this really large protein structure. And so if we think about it, this might be our big protein structure here. And the protein structure, the goal of that protein structure is to hold functional groups in that active site in the right position for catalysis to occur. All right, so with all this structure here is about holding functional groups in this active site in the right position. It might move them around during the reaction, right? We saw during hemoglobin that actually we had all this structure, but that structure moved around to change kind of how things were, to change the affinity of hemoglobin for oxygen depending on what was going on. And so then this would be our active site. Okay, and that's where the reaction is catalyzed. Okay, so in general, there's one you can think about enzyme catalysis as happening such that enzymes are going to catalyze reactions by having the right things at the right place in the right time. That's one way we can think about it, okay? I'm going to say providing or taking the right things at the right place at the right time. All right, so in other words, if a reaction needs, you get all these things in organic chemistry where maybe they needed a proton to catalyze it, right? You got some reaction, the reaction goes better if there's a proton attached. And so you dumped in a ton of acid, if you were doing this, so that there were protons around. Well, what the enzyme can do is when that reactant is ready for the proton, the enzyme puts the proton right there. It says, hey, here's a proton. Keep going. Okay? And we'll talk a little bit more about actual mechanisms for that. Yes?
You forgot place. At the right place.
Did I forget the right? Okay. That way there's some confused things. You catalyze by providing the right things at the right place at the right time. Okay? Thank you. All right, so that's one way to think about it, and we will see that. Another way to think about this is that enzymes lower the activation energy for the reaction. Okay, so enzymes, and these two aren't necessarily separate. Enzymes, let's say let's say offer a different reaction path that has a lower activation energy. And then let's say than the uncatalyzed reaction. Okay, so we can think about these in terms of free energy diagrams. So let's sort of put those two together here. And this is a picture out of a different book. I love this picture. I think it's really good. So let's say we have this enzyme called stickase. It breaks sticks. Okay, and you'll notice as we go and talk about enzymes, they end in ase, right, all the time. So we've got stickase. Stickase is going to break sticks. I'm looking for a good stick to almost break around here. But is there a pencil? Actually, a ruler will work. Should put my safety glasses on. Here we go. That'll be okay. I'll point it towards you guys. Okay, so if I was going to break this ruler, right, I'm going to put energy into that ruler to get it towards breaking, and I'm putting no energy in, I'm putting more energy in, I'm putting more energy in, I'm putting more energy in, okay? And just before it breaks, I'm going to have put the maximal energy in. You can see, right? Can you imagine that? Okay. So an enzyme is going to do the same thing. It is going to, it might actually bend it, but if nothing else, if this was going to break on its own in solution, right? This is it. This is a molecule in solution. It's flexing back and forth just to thermal energy, just because of thermal energy, and then occasionally one of these will get enough thermal energy that it will get to that point where it breaks. All right. What our enzyme is going to do if it's going to encourage this to break is this is going back and forth in terms of its thermal energy. It's going to go ahead and bind it when it's like this. And then maybe it has a little more thermal energy and then it does the break. Does that make sense? So it holds it near the break and then it's going to encourage it to do that. And that's what we see in this picture over here. Imagine how we have this stick. So what they've got is they've got this magnetic stick, but we know that everything's about charges. So we're going to change it to charges. So we could imagine that this stick was all positive charges, right? And so if we look at our example down here, if we have that stick and we can bind it when it is almost about to break, then it has a little thermal energy while it's there and then it breaks, right? We're holding it closer to where it's just about to break, and then it's going to speed up how fast it breaks. All right? So in terms of chemistry, what we've got here, and these are more complex than we're going to worry about. All right, so these reaction diagrams actually do have, you have to bind the stick, you have to bind it in its most energetic place where it's about to break, and then you go. There are possibly many steps like this, and I think they maybe talked about this a little bit in organic. Where you do your kinetic, your free energy diagrams, and depending on how many humps you have and where the humps and how high they are, it's thermodynamically favored maybe or kinetically favored. I can't remember. Product favored, reactive favored. I actually never taught it or learned it. So, but these things can be more complex. One thing that actually is related to the work I do for research is you could think about if this stick is going to be out here floating around in water, right? It's going to have water molecules on it. I have to take those water molecules off to make it bind. If it's positively charged, it sure as heck has lots of water molecules around it. Okay, but those water molecules actually have to physically come off of it. So it kind of has to be free of water molecules for a second, for a fraction of a second before it binds. And that'd be a high energy state. It's sitting in water without having lots of water stuck to it. Anyway, let's go ahead and look at, so what goes on here is when we have this enzyme has a active site that is complementary to our stick in its almost broken state. And here I'm going to put my positive charges on it. And then you imagine the enzyme has negative charges in this active site. Right? So one way you can think about it is now I'm binding it in its most active site or most active state just before it breaks, and that helps it break. Okay? But another way you can think about it is if I have positive charges and negative charges, what happens when I take a positive and negative charge and I bring them close together? What was that? They're attracted, and what happens to energy? Energy is released when I do that. Okay? Whenever I form bonds, these are sort of forming ionic bonds, energy is released. And so when we think about this active state here, we think about our transition state here. The energy of our transition state, if we suddenly bind things to it and release energy by binding them, we are lowering the transition state energy. Okay? So, whoops, I'm gonna plug this from the wall. Good, I didn't want it to dry. Okay, so do you see why we would lower the transition energy? Okay, we are releasing energy by binding to it. So that's great. Okay, so we lower the energy by binding to it. And you want to bind the enzyme active site to bind to that state about where the transition state needs to be complementary to the transition state. Does that make sense? Okay, so then here we have our example of what if our enzyme was instead, let's go ahead and put our positive and negative charges on it. What if our enzyme instead here bound and was complementary to the reactant or the substrate, okay, with the shape of the substrate, not in the transition state. Well, what's going to happen then? Well, if you look over here for this picture, the enzyme binds to that substrate. When it binds to the substrate, energy is released. And suddenly our energy is lower, right? And then if we want to get from our substrate to our product, energy is lower here. Now I'm going to go that far instead of going that far, right? This is the non-catalyzed energy from substrate to active site or transition state. And then this is my catalyzed. It's not catalyzed. I'm actually stabilizing the substrate by doing that. And now it's harder to get to product. You see that? All right, so what we need to take out of this is that couple concepts we want to take out of this. Enzymes catalyze reactions by binding to the transition state. Okay, another way of saying that isn't quite so binding, right? It doesn't get into the transition state and then your enzyme binds it. Maybe it's getting into it while it's going into the enzyme, but so their active sites are complementary to the transition state. Okay, when they bind, bonds of some sort are formed. Here we have kind of ionic bonds, right? Here we have, actually we have ionic bonds. It might be hydrogen bonds. It might be van der Waals interactions. Could be anything. When they bind, energy is released, lowering the activation barrier for the reaction. Or let's say lowering the transition state energy. Does that make sense? So I'd like you to be able to draw these activation energy diagrams, but I'm not really looking for you to draw. I mean, I'm going to put a lot of stuff on there, but as complicated a catalyzed path as we're seeing there. We're just going to do simple paths, and so if we think about the progress of reaction, this used to bother me when I was in college. I was like, what is that? I guess I've just learned to accept it. And we've got energy over here. So we've got, this is how far the reaction has gone. We've got energy over there. Let's say I've got a substrate that has an energy here, and I've got a reactant, I'm sorry, a product that has an energy there. I need to get between the two. This would perhaps be my uncatalyzed reaction path. All right, and then if I want you to draw a catalyzed path, I want to see something like this. All right, does that make sense? That path, this activation energy, the transition state here needs to be higher than this, right? Because that's the way it is. Otherwise, you never have the substrate at all. So there's our transition state right there. Here's our transition state for our uncatalyzed path. And looking at this, what is the energy, or what is the transition state energy for the forward reaction for the uncatalyzed path? I'm saying, what is the transition state energy? It's this, right? And so we can talk about that. We can go call about the delta G. Let's see. How are they doing it exactly? Okay. So we can talk about this being the delta G, or the activation energy for the forward reaction. Uncatalyzed. Does it make sense what I did there? This is the hill I have to get over to be a product. And then I'm gonna not do all of them because it's just gonna become an awful mess. But you need to be able to talk about this either way. What would this be? It would be the catalyzed. It would be the activation energy for the catalyzed reverse reaction. And so there's 4 things there, right? There's 2 forwards and 2 backs. Does that make sense? You should be comfortable with that sort of thing and be able to identify these. Notice this for the reverse reaction. What happened for the reverse reaction when this enzyme lowered the activation energy? Is the reverse reaction easier or harder, or it doesn't matter? Or it's actually easier when it's catalyzed, right? So before it's catalyzed, maybe I phrased that poorly. Before it's catalyzed, this is my activation energy for the reverse reaction. Once it's catalyzed, this is my activation energy. So when most enzymes are going to lower the activation energy for both the forward and the reverse reaction. So they're going to help us get to equilibrium, but they aren't going to drive the reaction. They're not going to affect which way the reaction goes, if that makes sense. So most enzymes lower the activation energy for both the forward and reverse reactions. So it's going to make both of them go faster. Enzymes do not, as catalysts, enzymes do not affect the driving force. They do not affect, they not drive us towards reactants or products. They don't affect the thermodynamics. So think about equilibrium.
Do they affect the rate of reaction?
Yes, they affect the rate of reaction. That's the whole point. They don't affect the thermodynamics equilibrium of the reaction. They increase the reaction rate. Okay. Does that make sense there? So kinetics, not thermodynamics. Yeah, Jacob.
Is there any relevance to examples of enzymes that don't affect forward and backwards equally?
So where you see that is if you have an enzyme that makes something that's then removed from solution. So let's say I have an enzyme and one of the products is a gas and it comes out of solution. You're not gonna speed it up in both directions. So that's where the most comes from. When you see a reaction and you've got an enzyme catalyze it, you should just assume that it's going to increase the reaction rate in both directions. But there are examples where it doesn't. Yes?
How do you see that on the graph?
How do you see that it increases the rate in both directions?
Mm-hmm.
That's a good question. So if we look at our graph here, we can actually, let's do it for the, let's just do it for both. So this is my, if my forward reaction uncatalyzed is here, right? And then this is my catalyzed. Do you agree that it goes faster in the forward direction? Okay. So then in the reverse direction, now we're going products to substrate, this is my uncatalyzed reaction, and this is my catalyzed reaction in terms of my activation energy. So do you agree it's going faster in the reverse direction?
Okay, yeah. I guess
It's a
I was
smaller
just
hill.
thinking less of like looking at the delta T and more looking at the activation energy specifically.
Okay.
Like the change in the activation
Right,
energy.
right.
Yeah.
So one other thing, so when we think about the equilibrium, what here determines, what that's on this graph determines what my equilibrium will be, what my equilibrium position will be on this reaction.
The relative energies in this.
Yeah, relative energies of the reactants and products. And so this would be when we talk about a delta G for our reaction, that's that. Okay, so that's the free energy change. So this reaction is spontaneous in which direction? Forward, substrates to products. And it's non-spontaneous in the opposite direction. And that's because of these, where the relative levels of these are. It has nothing to do with, these are kinetic, these two paths are kinetics, these are thermodynamics. Questions? Does that make sense? Okay. You want to be able to draw one of these and like annotate the various important parts. So activation energies, the free energy change for the reaction, all that sort of stuff. Okay. All right, so then let's talk about some interesting examples for enzymes, how enzymes catalyze reactions in terms of looking at some mechanisms. So enzymes can catalyze, we can come up with three kind of broad categories by ways that, or of ways that enzymes catalyze reactions. Let's say, yeah, three main mechanism categories. We'll just say that. Let's talk about, we're going to talk about acid base. All right, we can talk about covalent catalysis. All right, we could talk about metal catalysis. We'll just say call it metal catalysis. Or metal assisted catalysis. All right, and so an acid-base catalysis, a proton is given to the transition state or taken away from the transition state. Okay, covalent catalysis. The enzyme forms a covalent bond to the substrate at some point during the reaction. Okay, in metal-assisted catalysis, we have a metal doing something that we can't get from an amino acid, right? So this is like in hemoglobin, we needed an iron to reversibly bind oxygen. And so a metal provides functionality that can't be provided by an amino acid. Okay, in general questions in those, we're going to go over examples. All right. I want to point out these aren't mutually exclusive. You can have more than one of these going on in a reaction. All right, so here, what's an example of this? Let's go ahead and give it an example of acid-base catalysis. All right, so those of you in neuroscience or psychology will recognize this. You probably got in your biology courses too. But here I have a neuron, or a junction between neurons. Okay, so we have this gap junction where a signal is going to be passed on from one neuron to another. Sorry, I made a mess of this at this point. Okay, and so what happens here, we'll talk more about this later this semester, but we have an action potential that happens here. So we have these channels that open, they let sodium in, it changes the potential across the membrane. That causes more channels to open over here that let calcium in near the synaptic cleft. And what happens is there are little vesicles that contain acetylcholine. The acetylcholine is let out into the cleft and it goes and it binds to receptors on the next neuron. And that causes these receptors to open and let sodium and calcium in, which changes the potential, and then it causes these to open and those things go further, right? So this is what's going on here is we have this molecule called acetylcholine. It's right here, that needs to be released into the cleft to get the signal. That's what causes us our signal to go from this neuron to this neuron. All right. So then these channels open, but what would happen if the acetylcholine never went away? This acetylcholine binds this channel, the channel opens. If it never went away, that channel would just always stay open, right? We wouldn't be ready, we wouldn't reset so that we could send another signal. All right, and in fact, some of the nerve agents, neurotoxic agents that people use for chemical warfare, will actually cause these to just stay open. Okay, so we have to get rid of that acetylcholine, and it turns out the acetylcholine, this channel also has a functionality it's got an enzyme that will break down acetylcholine so that more needs to be made so we can reset for the next signal to come in. So what we have here we have acetylcholine. It will come in and it will bind to the active site of this enzyme, or we've got this part of the enzyme here. And what it's going to do is, so we've got acid-base chemistry going on. So in the active site, there's a serine residue. A histidine residue can come and take this hydrogen away from the serine residue. This is actually covalent catalysis. And then we're going to end up with a covalent bond attached. So this is covalent catalysis right here. That's an example here because we are making a covalent bond between our enzyme and the substrate. All right? Here, we're going to break bonds though, right? So we're going to do it with water, right? So now we come down here, water comes in. So in terms of breaking this bond, right? So that we kick the electrons up, the electrons come back down, and then this is going to pick up this proton as it leaves. You see that? Okay. So here now we have some acid-base catalysis. This histidine is acting as an acid. It was acting as a base before, but not really with the substrate. It's still acid-base catalysis, but it was acting with the enzyme as that. So now we have, this is acid-base catalysis right here. His acting as a acid. Right? It's going to provide the proton here that helps this leave. And this needs a proton because those things are pretty, it's a hydroxyl, that's a pK of like 15 right there. So it's going to try to leave. Then we have, what we have now is we still have that serine. We didn't break this serine bond to the rest of the substrate, right? So to regenerate the enzyme, we need to have this occur. And so I've got a water that's going to come in, and it's going to bind there and kick the electrons up. But as the water is coming in, for it to act as a good base here, what's the His acting as here? Acting as a base, right? This histidine right here, hopefully it's not too low for you guys, is coming in and it's taking this proton off so these electrons can come and make that attack. Yes?
And this is how acetylcholine is broken down.
Yes.
So that the channel closes.
So the channel closes, so we reset and can take another signal later.
Okay.
Okay, so we kick the electrons up here when this water attack, or it's actually hydroxyl-ish. Enzymes are kind of, so in organic chemistry, you would have pulled the proton off, it would have been a hydroxyl, the hydroxyl would have done the attack. In these enzymes, certainly in biochemistry, we draw a lot of arrows all moving at the same time. And I don't know how much of that is sloppy and how much of that is actually, you do get concerted things happening in enzymes where all the arrows are moving at the same time, but sometimes it's just sloppy too. So we're just going to accept it because we're not going to fix that. But so these come in, we're going to kick those electrons up, and then here now those electrons are going to come back down, and we're going to end up with a leaving group. But to get the leaving group to regenerate this, we need to put a proton back on the serine, right? This kicks down, this is leaving, but as it's leaving, it grabs that proton over here. It's kind of busy. Okay, but once again, the His is acting as an, what is it here?
Yes.
Acid. An acid. I guess I led it with the and too. But so you see here, I didn't actually talk about it up here. The His is acting as a base here because it's not really attacking that, but it's still the same sort of thing. So His is a base, it's an acid, it's a base, it's an acid as we run around this circle. Does that make sense? So we got acid-base chemistry and we had covalent catalysis. And this is very similar to what we see, and in fact, that is the mechanism we see for a serine protease. Okay? So I don't know if this is officially a serine protease or not. I'd have to look it up, but it certainly seems like it's a serine protease of some category. Okay. Questions on that? Yes.
So histidine is a residue off of the enzyme?
Yes.
Okay.
Histidine's a residue on the enzyme that is breaking down acetylcholine so the channel can close again.
Okay.
All right. So this is the active site of carbonic anhydrase. And it's going to catalyze the conversion of carbon dioxide to bicarbonate. All right, so I want to remind you of our bicarbonate buffer system. It's not going to be, it's more contextual than important for what we're doing right here. But remember, we've got our bicarbonate buffer system. This is so useful to know. It just comes up over and over again. You can use it. There's lots of things in nature and in your body that use it. Okay, we got our bicarbonate buffer system, and so for your blood to carry carbon dioxide, some of it's carried on your hemoglobin. We talked about that, right? It's a fair amount is carried on your hemoglobin, but a lot is also carried in your blood. But CO2 doesn't dissolve well in liquids. We know that, right? You open a soda can and all the carbon dioxide comes out once you put it at regular atmospheric pressure. Okay, so it needs to be carried as bicarbonate in your blood, but it actually doesn't. Your body's making carbon dioxide. Carbon dioxide bubbles would be really bad in your blood. Okay, and it can't. This reaction doesn't happen fast enough on its own. So we have carbonic anhydrase, which is one of the most speeds up this reaction like I want to say 10 to the 12. It's a huge number. This is one of the fastest. This speeds up the reaction more than like any other enzyme. It's like the record holder or really close to the top. So how does it actually catalyze this? Well, it's going to use a zinc. All right. So the zinc is coordinated in the enzyme by 3 histidine residues. In other words, those histidine nitrogens are in. They're holding the zinc in place. All right. And this zinc atom, what we're going to do is, let's see, where are we going to start? Let's start with carbon dioxide. All right. I guess that's 2, but I don't care. Let's start with the carbon dioxide comes in. Carbon dioxide comes in, and oh no, let's go. Well, yeah, we'll start here. All right. So carbon dioxide comes in, and the zinc is holding onto a hydroxide here. Okay. And that hydroxide is all ready to do that nucleophilic attack. Hydroxides like to do nucleophilic attacks. On this carbon dioxide, we kick the electrons up. This state is stable right here. We're trying to make a bicarbonate, right? And in fact, that is a bicarbonate. All right. And then another water comes in that not displaces the bicarbonate. So we just changed the carbon dioxide to a bicarbonate. And then now we have this water attached to the zinc. Well, we want to make it into hydroxide. What's the charge on hydroxide? Negative, right? So we have a negative right next to a positive if this proton leaves. So that's stabilized, right? So the purpose of this zinc is to help make it easier to make a hydroxide in this enzyme. Okay, so that's how the metal is catalyzing the reaction. So let's go ahead and write that real quick. So the zinc, okay, so the zinc ion in carbonic anhydrase binds to water and stabilizes we'll say the hydroxide, or let's say the loss of a proton to make hydroxide. Okay? And to really emphasize it, let's go ahead and just in that description, let's emphasize the charges. So I'm going to say negative, the zinc ion, 2+, okay? And then we'll just say the hydroxide attacks the carbon dioxide, carbon dioxide as a nucleophile. Questions on that? Okay, so you see how the metal's helping out here? And we actually do have amino acid side chains that can help make a water into hydroxide, right? We've seen the histidine do it, but this is how carbonic anhydrase evolved, right? Natural selection picked it. And also, it's super fast. So it may be that this is really that this is necessary. Yes.
Is this also a covalent and acid-base catalyst?
Well, we are getting a covalent, so we've got acid-base chemistry going on, right? And we do have, we could think about this as covalent, yeah, I guess. The substrate is so small here. I don't exactly think about it that way, but okay. So we are now ready to talk about serine protease, and we've already talked actually about, I really think the acetylcholinesterase. I'm going to have to look at that and look that up and see whether it is characterized as a serine protease, and I'm sure it is at least at some point. I'm not sure whether it has this full structure. So when we talk about serine proteases, first of all, let's talk about what they do. So serine proteases, and maybe it's not actually because it's not exactly breaking a peptide bond, is it? Right? The acetylcholinesterase. Let's go up here. Are we breaking a... We're not breaking a peptide bond there. So acetylcholinesterase, it's an esterase, but it's using a serine. So maybe it's a serine esterase. Anyway, serine proteases catalyze the hydrolysis of peptide bonds. And I gave you the original example of where we had a giant pile of these in the blood clotting cascade, but also, you know, you eat proteins and you've got several serine proteases in your digestive system that are going to break down those proteins into smaller chunks, okay? So those proteins are going to get broken up by those enzymes. So, and that's actually probably in your book, probably taught, the example in your book is probably chymotrypsin. Hold on.
It is, because that's what you have on the study guide.
Okay, so chymotrypsin is the one in your book. Trypsin, also a serine protease, lots of serine proteases. Okay, these enzymes have a catalytic triad of amino acid side chains. Okay, and so what those side chains are is we've got serine, and serine is like you saw previously, and we're going to go over this mechanism, although not in the next 2 minutes. So the serine is going to make a covalent bond to the substrate. Okay, we've got a histidine. Histidine is going to act as an acid and a base during the reaction. Okay, and then finally we have aspartate, and this is where we're going to start talking about something really cool in terms of these things. The aspartate is going to stabilize histidine in the positive state. So let's say the protonated state of histidine. Okay, and this is the really cool part. Okay, raising its pKa from 6 to 12, a full 6 points. Okay, so that's huge. When we look at these, so this is our aspartate, this is our histidine, and this is our serine. All right, we are going to raise the pKa of this, so it's really good at taking the proton off the serine. All right, it's still going to be able to give the proton back to other things. But remember, if you have your pKa up at 12, now any pH below 12, you're mostly protonated, right? Okay, we're going to go over the mechanism for this next time. I guess we've got a lot on Friday. We've got a lot of time here, but I will tell you ahead of time, this is the mechanism from your book. You need to memorize this mechanism, okay?
For quiz on Friday?
Not for Friday,
Okay.
but for the exam. Okay, we'll talk about it more on Friday.